Welcome to Study Pharma. Today we'll discuss an important topic which is drug discovery and clinical evaluation of new drugs. Historically, drugs were discovered by identifying the active ingredient from traditional remedies or by serendipitous discovery, which is an unplanned discovery of a new drug, just like the penicillin. Recently, scientists have been able to understand the shape of biological molecules at the atomic level and to use that knowledge to design drug candidates. Despite the advances in technology and understanding of biological systems, drug discovery is still a lengthy, time-consuming, expensive, and difficult process. Approximately 10 to 15 years of time is required from idea to a marketable drug. We are going to study this process in 4 steps. The step 1 would be drug discovery and target validation. Drug discovery is the process by which new candidate medications are discovered. This is where drug development companies choose a molecule, such as a gene or protein, to target with a drug. The phase during which the candidates or target of interest are selected. Target identification is to identify molecular targets that are involved in disease progression. Target validation is to prove that manipulating the molecular target can provide therapeutic benefit for patients. Lead discovery is the prototype drug around which further optimization is centered and focused. A lead compound is one that is believed to have potential to treat disease. This process also involves the identification of screening hits, medicinal chemistry, and optimization of those hits to increase the affinity, selectivity, efficacy or potency, metabolic stability, and oral bioavailability. Once a compound that fulfills all of these requirements has been identified, it will begin the process of drug development. This is where the drug developer will confirm that the molecule is indeed involved with the disease in question. After testing multiple drug molecules, the drug development company will choose those that have promise. Now let's move on to the step 2, the preclinical testing. Once a lead compound is found, the drug development begins with preclinical research to determine the safety and efficacy of drug. Preclinical trials test the new drug on non-human subjects for efficacy, toxicity, and pharmacokinetic information. It can be broadly divided into 2 subcomponents, the in vitro and in vivo testing. In vitro testing examines the drug molecules' interactions in test tubes and within the lab setting. In vivo testing involves testing the drug molecules on animal models and in other living cell cultures. Although efficacy is beginning to be established here, safety is paramount as the FDA will not let preclinical studies move into human trials without extensive data on safety. The following types of tests are performed. Screening tests. These are simple and rapidly performed tests to indicate presence or absence of a particular pharmacodynamic activity. For example, analgesic activity. The next test is on isolated organs, bacterial cultures, etc. The tests on animal models of human disease, such as Kendall seizures in rats, hypertensive rats, experimental tuberculosis in mouse. The general observational tests. It is performed either in the beginning or after detecting useful activity in screening test. The drug is injected into small groups of mice, which are observed for overt effects. The confirmatory tests and analogous activities. Compounds found active are taken up for detailed study by more elaborate tests, which confirm and characterize the activity. The mechanism of action. Attempts are made to find out the action of drugs. Pharmacokinetics. The absorption, tissue distribution, metabolism, excretion, volume of distribution, and half-life of drugs are quantified. Toxicity tests. They aim to determine the safety of the compound. They can be divided into the acute, subacute, and chronic toxicity. The acute toxicity, single doses are given to small groups of animals that are observed for overt effects and mortality for 1 to 3 days. Subacute toxicity, repeated doses are given for 2 to 12 weeks. Chronic toxicity, the drug is given for 6 to 12 months and effects are studied as in subacute toxicity. Let's move on to the step 3 now. Investigational New Drug Application filing, or the IND filing. An IND is submitted after the sponsor determines that the proposed drug is reasonably safe for initial use in humans and that it shows sufficient promise as the treatment to justify commercial development. The FDA reviews the IND application and decides whether it is safe for the company to progress to the next stage. This is the point where the FDA will scrutinize the results from the preclinical testing, look at the side effects and other safety features of an experimental drug. Examine the drug's chemical structure and how it's believed to work, and take its first look at the manufacturing process of the drug. If the FDA approves a drug developer's IND, then it can move on to the human trials. Now, the step 4, clinical trials. These are tested on humans. A clinical trial is only done when there is a good reason to believe that a new test or treatment may improve the care of patients. It is further divided as phase 0, 1, 2, 3, and 4. Let's start with the phase 0. It is done with a very small number of people, usually fewer than 15. It aims to learn how a drug is processed in the body and how it affects the body. In the phase 1, human pharmacology and safety, the first human administration of the drug is carried out by qualified clinical pharmacologists or trained physicians in a setting where all vital functions are monitored and emergency facilities are available. This aims to find the best dose of a new drug with fewest side effects. Doctors start by giving very low doses of drug to a few patients. Higher doses are given to other patients until side effects become too severe or the desired effect is seen. This study is open label, in which the trial participants are not withdrawn of information about the trial. Phase 2, therapeutic exploration and dose ranging. If a drug is found to be safe enough, It can be tested in a phase 2 clinical trial, which further assesses safety as well as if a drug works. This is conducted by physicians who are trained as clinical investigators on 100 to 400 patients selected according to a specific criteria. The primary aim is establishment of therapeutic efficacy, dose range, and ceiling effect in a controlled setting. The study may be blinded or open label and is generally carried out at 2 to 4 centers. Phase 3. Therapeutic confirmation or comparison. If a drug is found to work, it can be tested in a phase 3 clinical trial. It is used to compare a new drug to the standard of care drug. These trials are randomized and conducted on a larger patient population from 500 to 3,000 by several physicians at many centers. Every patient in a phase 3 study is watched closely. The study will be stopped early if the side effects of the new drug are too severe or if one group has much better results. Roughly 25 to 30% of medications move on to phase 4. The FDA usually requires a phase 3 clinical trial before approving a new medication. Due to large number of participants and longer duration, rare and long-term side effects are more likely to show up during this phase. A new drug application (NDA) is submitted to the licensing authority who, if convinced, give marketing permission. The phase 4 is the post-marketing surveillance or studies. This happens after the FDA has approved the medication. This phase involves thousands of participants and can last for many years. After the drug has been marketed for general use from practicing physicians, data are collected on a structured pro forma about the efficacy, acceptability, and adverse effects of the drugs. Uncommon adverse effects, or those that occur only after long-term use and unsuspected drug interactions, are detected at this stage. Further therapeutic trials involving special groups like children, elderly, pregnant or lactating women, patients with renal or hepatic diseases, etc. are conducted.